My name is Hartmut Geiger. I'm Institute of Molecular Medicine in Ulm, Germany, but have been also a long time at Cincinnati Children's Hospital Medical Center. So the initial research started actually in Cincinnati. And what I'm going to talk about today is stem cell rejuvenation by targeting CDC42. So that's not new to you, but I think we should always remember that we in science and research should identify molecular and cellular mechanisms of aging that cause aging-associated tissue dysfunction and diseases. And in our case, stem cell aging might be one of the underlying causes for tissue attrition diseases, cancers, in systems that rely heavily on stem cell activity in the adults, like skin and intestine, and hematopoiesis. I'll talk primarily about hematopoiesis today. If we know the mechanisms, we could develop innovative therapies for healthy aging. Very simple, but I think it's always important. So in the hematopoietic field, again, we work on, it's a stem cell field. And on top of this hierarchy, it's the stem cell hierarchy, we have these hematopoietic stem cells. And hematopoiesis is usually located in the bone marrow, as you can see. And in this bone marrow, we have a very rare population of these hematopoietic stem cells, 1 in 100,000. They self-renew there, they do multilineage differentiation via symmetric and asymmetric division. Then they make these progenitor cells, they're more frequent, but they have this decreased self-renewal, increased differentiation potential. And then these hematopoietic stem cells make all mature blood cells. And that includes all immune cells. We should not forget that stem cells make B cells, T cells, myeloid cells, NK cells, and so forth. So it's not just erythrocyte and other cells, it's the whole immune system. Good. Now I also need to introduce our favorite animal. It's a small road GTPase called CDC42. You probably heard about other road GTPases like RAS and so forth. And GTPases that are defined by what they bind and not what they do in the sense by a depressant. So only when they bind GTP, another small molecule, then they can interact with effector proteins. And then in the case of CDC42 is responsible for cytoskeletal reorganization, membrane trafficking, migration, adhesion, and many more things, which I also now refer to that is order within a cell. And the amount of GTP on CDC42 is regulated by factors that exchange GDP via GTP, and also factors that help hydrolyzing the GTP so that it becomes GTP again. And this is all regulated by many ligand receptors inside and outside the cell. So CDC42 is a good integration point of a lot of information. It'll be translated in a certain level of GTP bound, and then certain decisions in the cell will be made with respect to order. Now, a long time ago, a colleague of mine figured out that CDC42 activity is elevated in multiple tissues of mice. And we do that with the analysis with a pull-down assay. Again, it's not the expression level, it's the activity of the protein. So you have an effector protein that is able to bind to the GTP-bound form of CDC42. You can pull that out of a protein lysate, and then you develop it on a regular Western blot with a simple anti-CDC42 antibody. But as you pull it out with an effector protein, you know it has been GTP bound. This is the upper band here. And then you compare it to the total amount of CDC42 in the protein lysate of cells. It's also normal anti-CDC42 Western blot. And then you normalize it to a standard gene gap DH. And what you can see is that the activity of this CDC42 is elevated in almost all tissues that we studied so far. Some more, some less, like heart is high, liver is high, spleen, kidney. And I can also tell you that all hematopoietic cells show elevated activity. Intestinal cells show intestinal stem cells show elevated activity, and so forth. So the elevated activity of CDC4 is, I think, a novel and a likely universal hallmark of aging in stem cells and also other tissues. So Now, I need to go one back. So we could show that if you have a genetic tool to significantly elevate CDC42 activity in young animals, these animals clearly undergo premature aging. And we could also show that stem cells that have these hematopoietic stem cells that have this elevated level already in young animals clearly show signs of aging. So we know that it's not just a marker, it's also causative for aging, this elevated CDC42 activity. And we found first in a large set of studies long time ago that, for example, osteopontin in stroma and osteoblasts in the hematopoietic niche or Wnt5a within stem cells all lead to this elevated activation of CDC42 upon aging. And what we also found, which is shown on this side, this leads to disorder with respect to standard proteins and epigenetic markers in stem cells, hematopoietic stem cells. So this is one hematopoietic stem cells. Blue is DAPI, the nucleus. The green one is tubulin, and the red one is a histone modification, histone 4 acylated on lysine 16, that in young stem cells show a polar order distribution. Upon aging, this order is gone. So you have tubulin everywhere, you have the histone marker everywhere, and so forth. This is all caused by the elevated level of GTP of CDC42. And we have shown that a drug called casein, which stands for CDC42 activity-specific inhibitor, can reduce the activity of CDC42 by disrupting the loading of new GTP onto CDC42. And it does not affect other small road GTPases, so it's very specific for CDC42. What the drug is doing, it returns, for example, this apolar situation to stem cells into a polar one, and it also changes then the epigenetic status of stem cells and the hematopoietic stem cell function. So as we publish now, it clearly rejuvenates the function of hematopoietic stem cells if the drug has been given ex vivo to stem cells and retransplanted. So that's the whole summary. Good. Then we figured out that it can also, when given to animals in vivo, for example, alter hair follicle aging. So you can clearly show that if given to animals in vivo, old animals, you have in part here regrowth of hair follicle. Aged mice don't like to grow hairs. We could also show and publish that intestinal stem cell aging is driven by changes in CDC42 activity. If you give the drug to animals in vivo, aged ones, you can restore intestinal stem cell activity under stress situations. So you also attenuate or rejuvenate aging of intestinal stem cells. A collaborating group in Munich here has shown that casein, so if you target that, you can affect aging and stress of mesenchymal stem cells so that you can also start addressing stress and rejuvenation of the bone marrow niche and mesenchymal stem cells, also given if the drug is given in vivo. That's all published in these papers. So, good. That means CDC42 activity elevation seems to be a central driving force in aging of the mouse. And if you can generate or inhibit the activity with this drug, CDC42, casein, then we can restore stem cell function in aged animals to a young level on multiple types of stem cells. Now, a mouse is a mouse. Usually we're interested in humans. So we checked also the function of aged human hematopoietic stem cells and how they look like. So first of all, this is what I said. This is now bone marrow cells from aged human beings. So this is young. You see a tiny band of GTP, CDC42 GTP, and this is an aged human being. So you see a high level on it. This is now more data. We had a large cohort of young and old people. You see the age range 20 to 40 roughly, and 60 to 80. And you see clearly that, first of all, there's an increase in relative CDC42 activity upon aging. And as you anticipate from a heterogeneous human population, you see also very heterogeneous difference in this increase. The interesting part is that the more CDC42 you have activity, you seem to change also the frequency in stem cells. It's going a little bit up, as we see, for example, also in the mouse, that you have more stem cells. And we have published that's probably due to a more symmetric division of these old stem cells while they lose their function. That's the interesting one. They have more, but they reduce their function. And we could also show that this polarity part I described for the mouse stem cells, in this case for the protein CDC42, the young ones, they are polar, and the aged ones, they are apolar. I'll come to that in a minute. Now, if you give casein back to these aged stem cells, you start to repolarize them. Again, see here, only 20% are polarized. And then if you titrate back casein, you get this up to 60% are polarized. It goes almost to the level of these young stem cells. And then we did xenotransplantation experiments to also test the function of these rejuvenated aged human stem cells in a transplant setting. The transplanted setting has some limitations because it's a xenotransplantation. So some of the function of the stem cells might not be read out perfectly. But we could see is that you have, interestingly, a bit higher contribution of these aged stem cells, might be to this artificial more cell renewal that in vivo will not lead to better stem cells in a real human being, as you have shown in the mouse. But you can see that if these aged stem cells get casein, they perform again in this assay similar to young ones. And again, because the assay is not really cool, we then started to do single cell sequencing with SMART-Seq2 and hematopoietic stem cells from a young and aged human beings. It's done by Medan Mulav, a collaborator at Ulm University, and compare young and aged stem cells first, and then the ones treated with casein. So the first thing we could do is now, in contrary to the mouse, it looks like that you can easily separate the expression profile of young and aged stem cells of humans. So you can have profiles with top 5 predictors in a PCA analysis. They can clearly tell you this is an aged stem cell and this is a young stem cell, meaning from an aged person, from a young person. And this is already the data set we used after we used AI to establish it. So really good now in separating them. And the AI training has not seen any of the stem cells treated with casein. Now, this is on the right side, you see here that we have RNA expression profiles now from donor A and B. They're all elderly donors. So this is material from 70-year-old people. And you can see donor B has only aged stem cells in the analysis. Donor A, although aged, has stem cells that are clearly aged, but also has some of the young ones. But what's more important now, if you give precisely these cells, donor cells, casein, the rejuvenation aging to donor B, then you see that all of a sudden the stem cells are to 60% young and only 40% remain aged. And even in a donor A, where you have already still some young stem cells, this goes down to 70% now being young and 30% remaining aged. We have some idea why these ones remain aged. They might not be real stem cells anymore. So we can also rejuvenate on the gene expression level with the drug human hematopoietic stem cells. Good. Now we go back to mouse. If all the functions I told you about are really true, then it would be also interesting to test whether the inhibition of CDC42 GTP with the drug in animals in vivo can modify lifespan. So we injected 75-week-old animals from day 1 to day 4, just 4 days in a row with 20 milligrams per kg, and just one injection every 24 hours. Then we took later on cytokine arrays, and then we also did at day 5 a peripheral blood analysis for methylation aging, as you're familiar with that, and we also measured survival. So now this clearly shows you that animals that received casein, the drug here, had a significant increase in median lifespan and maximum lifespan, which is around 10%, but it's the higher level of interventions that are out there. And I think we're still the only ones that can give it late in life, only for their 4 days. They never see the drug again. They have a long-term memory of what they have seen and translated in lifespan extension. And you're all familiar with biological clocks. This is done work by Wolfgang Wagner. So we use the Wagner clock for chronological aging, biological aging. So in the controls, the age controls here, you see that the chronological age and the biological age determined by the methylation clock of Wolfgang are identical. Now, in the animals that got treated with casein, again, 35 days after the treatment, that's roughly here, you already see that these animals, while still chronologically aged, have a much more reduced biological age by this methylation clock. So this is not a drug that is verified on the biological aging clock level to represent this extended lifespan. Good. So summary 1, murine in human stem cells aging is driven by elevated activity of CDC42. Murine in human stem cells can be rejuvenated by inhibition of CDC42 via casein. And we see immune rejuvenation come to that. Casein extends lifespan and attenuates aging in vivo and affects most likely multiple stem cells in vivo, not only hematopoietic ones. Good. So this is what I think what aging is. As we said, if you age, you start having disorder in your stem cells, measured in our case by epigenetic disorder, but we have also other tools to measure other types of disorder distribution. And this is young rejuvenation in the cell. And such a concept could be the main driving force of aging. Good. The last 10 minutes, I would quickly go on what is the consequence of aging hematopoietic stem cell, what's going wrong in hematopoiesis. So as you know, as I said, all the hematopoietic stem cells drive the immune system. There's large discussion on whether the immune system is one of the main limiting factors for healthy aging and longevity. And what we call now aging-associated immune remodeling, that there's an increased susceptibility to infections, responsiveness to vaccines is decreased, and you have increase of diseases and cancers. And we think that has all to do with aging of hematopoietic stem cells. And we came up with a system to test that. It's a simple immunization schedule we do in mice. So we immunize with hepatitis B core antigen because people in a DNA vaccine, because people like that in the animal. And then later on, we measure the antibody, the T cell response by by specific peptide-loaded H2K dimers, which bind on the cognate T cell receptor that would go against the hepatitis core antigen. So we can measure immunization response in this direction in the flow cytometry chart, and we measure that in that case on CD8 T cells. And what you can see is simply in young and aged animals, you immunize them. There's less immunization in aged animals. There's no surprise. Now we can measure that in animals that got be transplanted with young stem cells and transplanted with old stem cells. We could show that these make a young immune system in these young recipients, and in this situation, you have an old immune system in old recipients. And you can also see that the age of the immune system drives the immunization response. So although you have young animals here with an aged immune system, they don't respond very well to the immunization. And that allows us to test one now if you transplant these old stem cells but they have been rejuvenated with casein, in this case ex vivo, what would be the immune response? And what you can see here nicely, this is, for example, done in spleen, as shown before. You have a very small or low immune response to the vaccination in animals that got old stem cells. Now, these animals got the same stem cells, but they were rejuvenated with casein. You're back to the levels you see in young ones. And it's even more extreme in the liver, where you have really a strong immune response of these rejuvenated stem cells. And what's even more interesting is, so casein can restore immune responses via rejuvenation of hematopoietic stem cells. And now we also looked at transcription profiles of naive CD4 T cells and CD19 B cells in this transplantation situation. So these B cells and T cells have been derived from the transplanted stem cells. And the animals only got stem cells. They did not get T cells and B cells. So they have been through a long differentiation process coming from the stem cells. And what you can see now is that these very differentiated cells that come from these stem cells show expression profiles that are on average pretty close to young ones, almost perfect for the T cells and almost perfect for the B cells, but they're very distinct from the B cells that are derived, for example, from aged stem cells. But the only time they have seen the drug was before they were transplanted, the stem cells into the animals. So what we have is a very long memory effect of the drug treatment on stem cells and likely also other cells currently think via epigenetic mechanisms. So there's a strong memory effect if you know a system for drug treatment. Good. The last part I want to talk about is that we also figured out very recently is data of a collaborator, Yi Cheng in Cincinnati, that the drug or CDC42 activity attenuation also directly acts on Tregs and can boost T effector cells. So this is a kind of aging or exhaustion in which we then can use them for immunotherapy against cancer. So here you have a tumor volume. In this case, it's a colon cancer model system, a very established one in mice. And if they usually have a tumor model, you give them the tumor and then the tumor grows in these animals. If you treat these animals with casein, now multiple times each day or once, then the tumor is not growing that fast. That goes along with what you have. You have Tregs that become more unstable, means they express more interferon gamma. And you can have T effector cells that increase in numbers, that both you see in these animals. And other experiments showed that this difference is really due to changes in CDC42 activity. And the best part is now that you can also combine that with checkpoint inhibition, which is currently the standard therapy in some of these cancers. And you can see that you have, first of all, tumors growing when you don't do anything. Then you have casein or a CDC42 attenuator in the animals. And you clearly see that the tumor growth is strongly inhibited. It's not fully, it's strongly delayed. Similar to what you see in the standard anti-PD-1 therapy. Now, if you combine both in this model system, there's almost no tumor growth. Actually, it's a shrinking of tumor, and it's very rare also in animal models. Good. So that means we have immune rejuvenation, attenuation of immune exhaustion via rejuvenation of stem cells, and casein, so attenuation of CDC42 activity, can also directly act on exhausted HT cells to provide and further enhance cancer immunotherapy. With that, again, I want to mention one more time, I think that from our research now angle, this is what the core root of aging is. You go from order to disorder, and the simple trick is to restore order, and that you solve a lot of problems of aging of cells.